We're going to look at the structure of DNA. So let's first start with what's the point of DNA? What's the role of DNA in a person's body? So DNA its job is to store. It stores genetic information. It is going to store the information that we have been studying for all of those traits. So if the flower is going to be purple, the DNA is going to store that information to tell that flower that it is going to be purple. It then has to copy itself. Every time a cell divides, it must take an exact copy of the purple pigment gene. So DNA must be able to copy itself properly. And then DNA also needs to express itself. If DNA is just storing the information and then copying the information, but isn't actually expressing it in any way, you're not going to get that purple flower. The DNA needs to put that information to work, and the cells will use the instructions for the DNA to then make that flower purple. So DNA, how does it look? So DNA is made up of a nucleotide structure. Okay, so make sure, there we go. We have a base, we have a 5-carbon sugar, and we have what's called a phosphate group. So the DNA is made up of those nucleotides that are joined into long strands by what are called covalent bonds. The nucleotide acids are made, these acids are the building blocks of our nucleotides, okay? So that is how basically the structure. This isn't what DNA looks like. It starts to form and have all of these different bases. So there are 4 different bases for DNA. There's adenine, guanine, cytosine, and thymine. Scientists will often refer to these 4 different bases just by using the letters A, G, C, and T. So that's how we're going to do it in this class as well. We're going to just refer to it most of the time with the letters A, G, C, and T. Okay, so the bases are actually joined together by these covalent bonds here to connect to our 5-carbon sugars structure. So this is the structure of DNA. So you have your 1 nucleotide, your phosphate group, your 5-carbon sugar, and your base. That makes up 1 nucleotide. Well, 1 nucleotide isn't going to make up DNA. You have to connect it to many others. So you have these covalent bonds here that actually connect our nucleotide to our next nucleotide. Okay? I also want to point out that if you look here, how does adenine look different from cytosine or thymine? Adenine and guanine look a little different from cytosine and thymine. So the next thing that we're going to look at is this individual, a scientist named Chargaff, came up with this rule, and that helped to determine the structure of DNA. And he determined that every A equals 1 T. So if you have 10 A's, 10 adenines, you also have 10 thymines. If you have 10 guanines, you have 10 cytosines. And this is really important, especially when we get to the structure of DNA and how it actually forms, because these 2 are going to link up together, just like adenine and thymine are going to link up together. Okay? So we know, everyone's seen DNA, we know that it forms the kind of spiral. And everything we've been looking at so far has just been one side. Well, how did we get to the point of having this kind of spiral pattern? Well, it is called a helix, and this individual here is Rosalind Franklin, and she's the one that really helped us discover that DNA is actually a double helix. It's 2 strands of a molecule, and the bases are near the center of the molecule. And she helped us discover this by taking an X-ray of DNA, and she noticed that, oh, it's very kind of dark right here in the middle. So that is our base. And she didn't know exactly what this meant, but she did know that it showed us that DNA and this X of this X-ray suggested that there were two strands to the molecule compared to what we thought, which was just one strand. Okay, there we go. So Rosalind Franklin's work was then taken by Watson and Crick, and it was these two that actually put together the information to determine how exactly the two strands of the nucleotide sequences wound around each other to form this kind of twisted ladder. So there's a twisted ladder. We will look at a twisted ladder kind of visual of this when we get back. But Watson and Crick pointed out that the kind of base of the ladder were the base pairs. So that adenine, guanine, cytosine, and thymine. Okay? And we're going to be drawing a picture of this in a little bit. So here's our double helix. And one thing that I really want to point out is that our double helix isn't just both of them going up, directly up. So you can see here this arrow is pointing up, and this arrow down here, if that'll go away, is pointing down. The two strands of the DNA actually run in opposite directions, but then they connect at the center. And the running in opposite directions is what kind of gives it this spiral, okay? And this is going to be really important when we talk about building DNA. This is called anti-parallel, and that's going to be really important when we continue. So there is one last thing that I need to point out. These hydrogen bonds. So we had been looking just at one side. We had been looking at our nitrogenous bases. And we saw that cytosine connected to this 5-carbon sugar connected to a phosphate group. Okay, but they need to actually connect to each other. So this is where it's really important that guanine always connects to cytosine. Adenine always connects to thymine. So that is formed by hydrogen bonds. We're not going to go super in depth with hydrogen bonds, but you just need to know that it is formed by hydrogen bonds that hold it together. It's basically the glue of your DNA. So, as we said, the two strands of DNA are held together by the hydrogen bonds between the nitrogenous bases, adenine and thymine, and between guanine and cytosine. You are never going to get guanine connected to adenine. You're never going to get cytosine connected to thymine. You're always going to have G and C and A and T.